sĩ vui lòng chú ý lắng nghe. Lời đầu tiên, thay mặt ban tổ chức xin được gửi lời chào trân trọng nhất đến toàn thể các quý vị bác sĩ đã dành thời gian tham dự vào buổi hội thảo trực tuyến từ Brain School chuyên đề cập nhật các thông tin mới nhất trong hỗ trợ sinh sản, được phối hợp tổ chức bởi Bệnh viện Phụ sản Thiện An và Văn phòng đại diện Việt Nam lúc 14 giờ hôm nay ngày 4 tháng 12 năm 2020. Tham dự buổi hội thảo ngày hôm nay, chúng tôi xin được trân trọng giới thiệu sự hiển diện của ban chủ tọa và báo cáo viên. Chủ tọa tại Việt Nam, xin được trân trọng giới thiệu anh hùng lao động, nhà giáo nhân dân, giáo sư tiến sĩ bác sĩ Nguyễn Viết Tiến, nguyên Thứ trưởng Thường trực Bộ Y tế, nguyên Giám đốc Bệnh viện Phụ sản Trung ương, Chủ tịch Hội Phụ sản Việt Nam VAGO, trưởng Bộ môn Phụ sản Trường Đại học Y Hà Nội, Cố vấn cao cấp Bệnh viện Phụ sản Thiện An. Các báo cáo viên nước ngoài với tiến sĩ Pedro Barry, chuyên gia nội tiết sinh sản, Chủ tịch danh dự Hiệp hội Sinh sản Tây Ban Nha, tiến sĩ Paul Piet, chuyên gia dược lâm sàng Đại học VUB Bỉ, phó giáo sư Sandro Zeri, chuyên gia sản phụ khoa Đại học Perugia Ý, giáo sư Umberto Simeoni, cựu Chủ tịch Hiệp hội Chu sinh châu Âu, Đại học Lausanne, Thụy Sĩ. Kính thưa quý vị bác sĩ, để mở đầu buổi hội thảo trực tuyến ngày hôm nay, xin trân trọng giới thiệu anh hùng lao động, nhà giáo nhân dân, giáo sư tiến sĩ bác sĩ Nguyễn Viết Tiến, nguyên Thứ trưởng Thường trực Bộ Y tế, nguyên Giám đốc Bệnh viện Phụ sản Trung ương, Chủ tịch Hội Phụ sản Việt Nam VAGO, trưởng Bộ môn Phụ sản Trường Đại học Y Hà Nội, Cố vấn cao cấp Bệnh viện Phụ sản Thiện An có đôi lời phát biểu. Xin được trân trọng kính mời giáo sư ạ.
Vâng, kính chào tất cả các quý vị khách quý, toàn thể các anh chị em chuyên ngành lĩnh vực hỗ trợ sinh sản của Việt Nam. Trước hết cho phép tôi thay mặt tất cả những người tham dự về phía Việt Nam xin trân trọng cảm ơn đại diện của hãng Beijing Healthcare đã tổ chức cái buổi hội thảo hết sức là quan trọng ngày hôm nay, và cũng xin cảm ơn tiến sĩ Pedro Barry, tiến sĩ Paul Piet, phó Gali và giáo sư Umberto Simeoni là những nhà học, là những nhà chuyên môn rất là nổi tiếng ở nước châu Âu, ở nước Tây Ban Nha, Bỉ, Ý và Thụy Sĩ. Và cũng xin và rất là vui trong cái buổi hội thảo hôm nay có bác sĩ Sa, người Sa là đến từ nước Pháp, người cũng rất là thân quen với anh em chúng ta, và tôi cũng đã rất may mắn đã có lần đến chơi thăm nhà của bác sĩ Sa và cùng đã trò chuyện, cùng có những buổi rất là đầy ý nghĩa ở nước Cộng hòa Pháp. Và như chúng ta biết, cái hội của chúng ta hiện nay cũng phát triển rất là đông, nhiều các thành viên trong lĩnh vực phụ sản, khoa và sơ sinh nói chung, đặc biệt trong lĩnh vực hỗ trợ sinh sản. Và chúng ta cũng đã có biết được rất nhiều các kỹ thuật, nhưng chúng ta cũng thấy để mà làm ra được một cái thai nhi thì cũng không phải là quá khó, nhưng chúng ta làm càng nhiều, làm càng sâu, chúng ta mới thấy cả một công trình để làm sao mới giữ được cái thai, để đảm bảo cho được đứa bé về nhà an toàn, khỏe mạnh, mang lại hạnh phúc tràn trề cho gia đình của cháu bé. Quả thật đấy là một điều cũng rất là khó. Và cũng mặc dù tỷ lệ thành công của khi pregnancy đấy, khi có thai thì tỷ lệ cao, nhưng mà tỷ lệ baby take home vẫn đang còn rất là khiêm tốn, và chúng ta phải cố gắng rất là nhiều. Và mặc dù chúng ta cố gắng nhiều, cũng áp dụng nhiều kỹ thuật, nhiều phương pháp, nhiều thuốc men điều trị, nhưng kết quả cũng chưa đáp ứng được cái mong muốn của chúng ta cũng như bản thân của các cái bố mẹ. Do vậy mà hôm nay thì chúng ta tập trung vào cái chủ đề để làm sao duy trì, giữ được cái thai cho đến cái giai đoạn mà khi đứa bé sinh ra có khả năng sống một cách độc lập và ít để lại những dị tật, những cái tật nguyền, thì đấy là vô cùng quý giá và cũng rất là khó khăn. Thì với cái tinh thần như vậy, hôm nay sẽ có 4 báo cáo viên có rất giàu kinh nghiệm của châu Âu, đến từ đất nước Tây Ban Nha, Ý, Thụy Sĩ và Bỉ, và chúng ta sẽ lần lượt nghe được các cái báo cáo viên trình bày, và sau đây chúng ta sẽ có những câu hỏi thắc mắc và chúng ta sẽ trao đi đổi lại, và cuối buổi hội thảo chúng ta cũng sẽ có những cái ghi nhận lại, nốt lại những cái điểm gì là quan trọng, và những cái kiến thức đấy thì sẽ rất là bổ ích cho chúng ta trong hành nghề, trong công tác hàng ngày mà chúng ta phải đối mặt với những khó khăn trong việc giữ thai. Và và buổi hội thảo của ta sẽ bắt đầu ngay sau đây. Xin cảm ơn.
Dạ vâng ạ, sau đây sẽ là bài chia sẻ của các báo cáo viên nước ngoài được trình bày. Quý bác sĩ nghe dịch thuật song song sang tiếng Việt, vui lòng làm theo hướng dẫn trên màn hình slide. Bác sĩ chọn mục Language Interpretation, tức mục thông dịch ngôn ngữ. Chọn tiết mục Vietnamese, tức tiếng Việt, để nghe thông dịch tiếng Việt. Trong trường hợp cần hỗ trợ, xin liên hệ ban tổ chức của chúng tôi. Xin trân trọng cảm ơn quý bác sĩ. Mở đầu hội thảo là bài báo cáo với chủ đề sự phù hợp của việc theo dõi nồng độ progesterone trong chu kỳ chuyển phôi trữ của tiến sĩ Pero Paris, chuyên gia nội tiết sinh sản, chủ tịch danh dự Hiệp hội Sinh sản Tây Ban Nha. Xin kính mời quý vị bác sĩ chú ý lắng nghe.
Thank you very much, Sandro, and thank you, Giancarlo, for inviting me to this very interesting course, virtual course, and thank you for the praise school initiative in this world where unfortunately we have no opportunity to meet face to face and discuss scientific issues. At least we have we very thankful to this academic initiative such as yours, Praise School. We have the opportunity to learn, to teach, to discuss, and to meet. And then I think we have to adapt to the current situation and to do our best in this scientific, in this epidemiological situation that affects all over the world. It's now my pleasure to start my presentation. I will try to just a second. Can you see my slides?
No, not yet, Professor.
Not yet. Just a second. I will share.
No, aspetta. So, okay.
And now?
Yes, now we can see the PowerPoint. Can you put it in presentation mode? Good.
Okay, then.
Thank you very much.
My topic is to discuss with you the clinical importance of monitoring progesterone in ART. And I would like to start by presenting the outline I'm going to follow. I will present just brief messages of the importance of progesterone in fertility, the need of progesterone supplementation in ART, the different forms of progesterone administration that we currently do have, the increase of frozen embryo transfer cycles, the need for progesterone supplementation in this frozen embryo transfer cycle, and I will finally present the importance of low levels of progesterone in this frozen embryo transfer cycle. You know very well, and I would like to remind you the classical words published by Sappho, in which they, long time ago, they show the importance of progesterone in the luteal phase and in the early weeks of pregnancy. In what they show is that in patients in which lutectomy was performed early in the pregnancy, before 7 weeks of pregnancy, and miscarriage occurred in all the cases. When a lutectomy was performed beyond 8 weeks of pregnancy, the pregnancy continued. What does it mean? This means that this lutectomy was performed after the luteal placental transition, and the responsibility of maintaining the pregnancy was already in the placenta and not more in the corpus luteum. But if the lutectomy was performed before 7 weeks of pregnancy, and a supplementation of progesterone was administered to this patient, the pregnancy continued. So we were able to circumvent this lack of progesterone by giving it. And what's the role of progesterone in assisted reproduction and in human reproduction? Progesterone, as we know, is crucial for transformation the endometrium from proliferative to secretory endometrium and to induce endometrial receptivity. It's important to reach a status of myometrial cohesion that relax the uterus. It is important to induce an immunomodulation process, which is also crucial for the dialogue between the uterus and the embryo. And it's clear from the very beginning, we know that there is an important need of progesterone supplementation in assisted reproduction, both in IVF stimulated cycles and in frozen embryo transfer cycles as well, regardless if they were artificially prepared cycles or natural cycle frozen embryo transfer. And why? Because even in artificially prepared frozen embryo transfer cycle, there is no proposal. There is a clear need for progesterone supplementation, and even in cycles of frozen embryo transfer carried out in a natural cycle, there is most probably a luteal phase insufficiency that needs progesterone supplementation. We nowadays have different forms of progesterone administration. Mostly in Europe are vaginal natural progesterone or sub-Q progesterone administration. The effects and the benefits are similar. There are different publications related to the patient's preferences, some for vaginal progesterone, some for sub-Q progesterone, according to different parameters, the interference with sexual relationships, the feeling of dirtiness, etc. But both ways can be used, and even oral progesterone can be administered for what happened with frozen embryo transfer cycles that we are doing more and more in our house? Is frozen embryo transfer the future in the European? In fact, in my department, in the Instituto Universitario de Cios in Barcelona, last year, almost half of embryo transfer cycles carried out in our department have been frozen embryo transfers. And from year to year, there is an 20% increase, yearly increase of the rate of frozen embryo transfer cycles. And why we have this increase in the number of frozen embryo transfers? First, because thanks to the progress and the evolution of cryotechnology, and thanks especially to embryo vitrification, we have extremely reduced the rate of cancellation of frozen embryo transfer because no embryos survived the thawing process and no embryos were available for transfer. Nowadays, in our experience, this is less than 2% of the cases, and more than 95% of these patients undergoing a frozen embryo transfer, they do have an frozen embryo transfer cycle. And the other reason for this increase are, first, the set policy, the single embryo transfer policies that are applied more commonly in Europe and more in the US, and above all, the freeze-all strategies that we apply mainly in cases of prevention of ovarian hyperstimulation syndrome in cases of pre-implantation genetic testing requires deferred embryo transfer. In case of high progesterone levels in the periovulatory period that we know that they are harmful for the outcome of the cycle, and in cases of asynchrony between embryo and endometrium. And we know that in these cases, the pregnancy rate can be higher in frozen embryo transfer cycle. In cases of endometriosis, of risk of thromboembolic diseases, and in cases in which medical and elective fertility preservation should be applied. And of course, nowadays we have new ovarian stimulation protocols, some of them starting the stimulation during the luteal phase, and in this case, we will not have any receptive endometrium, and we will need to freeze the embryos and transfer them in a second artificial or natural. However, this following frozen embryo transfer does not provide a universal license to cheat. We do not have to freeze everything and to transfer in 100% of the cases the embryos later. But this frozen embryo transfer cycle after freeze-all would provide us good results, and we know from the literature that in cases of normal responder, applying the freeze-all strategy shows an increase in the live birth rate obtained compared with fresh embryo transfer cycle. And in case of high responders at risk of ovarian hyperstimulation syndrome, applying the freeze-all technique and segment and transferring the embryos in later frozen embryo transfer cycle provide a superior, a higher clinical pregnancy rate, live birth rate. In these cases, we need, as we have said, a progesterone supplementation. And even in cases of natural cycle, the patients with a good ovarian function, that if we can follow a natural cycle, you can see in this slide how giving extra progesterone to support the luteal phase improves clinical pregnancy rate, the live birth rate, and significantly reduces the miscarriage rate. So this is important even in natural cycle. We know that we can use either natural progesterone by oral or vaginal or rectal route, or cutaneous administration of progesterone to have a good, well-prepared artificial cycle. And Dominique Desigler established that by giving, for example, sub-Q progesterone, 25 milligrams per day, you can obtain secretory transformation of the endometrium in 100% of cases, and you can obtain full predecessor changes in more than 80% of the cases. But, and this is the point, what happens if the progesterone, the plasmatic progesterone levels are low before an embryo transfer in frozen embryo transfer cycle? And we know from the work from Elena Labarta, the work was done in the oocyte donation model. What Elena shows that when on the day of embryo transfer, progesterone level was below 9.2, the ongoing pregnancy rate achieved was significantly reduced compared to the pregnancy rate obtained when progesterone level was higher than 9. We analyzed and published our own material in the frozen embryo transfer cycle, measuring progesterone on the day before embryo transfer. And what Sofia Gaggiotti Madre published is that when our cutoff value was 10.6, when we were below this cutoff below 10.6, as you can see here, pregnancy rate was not affected, but miscarriage rate was reduced, significantly reduced when progesterone level was below 10, and live birth rate was significantly reduced as well. So we need to have a progesterone level above 10.6 before embryo. And the question was, could we reduce this miscarriage rate and can we increase the live birth rate in patients with low progesterone by giving more progesterone? And we analyzed the results obtained in frozen embryo transfer cycles of 115 euploid blastocyst frozen embryo transfer cycle. The preparation at that time was a bolus of generate albumin was given in most of the patients, and the standard artificial cycle was accomplished by giving patient 6 milligrams per day of lanerate estradiol and 200 milligrams of progesterone every 8 hours. And we faced two different situations. In patients where the progesterone level before embryo transfer was above 10.6 nanograms, we consider normal progesterone, and we proceed with thawing the embryo and transferring it. When progesterone was below this cutoff, we give extra doses of progesterone. In this study, was administered 25 milligrams of subcutaneous progesterone daily, but in other places we were given extra doses of vaginal progesterone, and we measured progesterone 1 day after. If progesterone level has increased above this 10.6 cutoff value, we consider that it was the group of progesterone rescue, and we could proceed with thawing and transferring. But in spite of giving more progesterone, the progesterone level was still below 10.6. We consider that it was not a good cycle, and we decided to cancel the cycle. Okay? So we can see here the figures, the numbers, and you can see how in some cases we succeed in increasing progesterone level by giving extra doses of progesterone. As you can see here, no difference in terms of pregnancy and miscarriage comparing the group of normal progesterone and the group of progesterone rescue. So you can act, you can do something on the day of before embryo transfer to improve the outcome in cases of low progesterone levels. Another point is, when should we measure progesterone? Because we know the classical words of Marco Filippori, that he showed that measuring progesterone, plasmatic progesterone, in the early luteal phase, mid luteal phase, and late luteal phase, that in mid and late luteal phase there were huge fluctuations in progesterone levels. And the question was that should we rely on a single value of progesterone to assess the adequate progesterone secretion? Our answer to this question is no. And we have published, Iñaki Gonzalez from our group published recently Human Reproduction, measuring progesterone levels in oocyte donors undergoing an artificial cycle, and measuring progesterone at different times during the day. What Iñaki shows, he showed that there was a 40% decline in progesterone levels during the day, from 8 o'clock in the morning to 8 o'clock in the evening. And we also analyzed which factors were associated with this low progesterone levels the day before embryo transfer in the patient received standard regime of estradiol progesterone supplementation. And it's interesting to know that the main factors that were significantly associated these low progesterone levels were the age of the patient, the weight of the patient, and the time in which progesterone was measured. Obese patients, old patients, were more prone to present low levels of progesterone, and we suggest to measure progesterone always at the same time of the day to be able to give value to the results of it. And the most important factor was the previous low progesterone level in a previous cycle. So if a patient has presented with low progesterone in a cycle, she will probably do the same in a subsequent cycle. So we have to advance the situation by giving the second cycle higher doses of progesterone. And we have also observed this phenomenon even in natural frozen embryo transfer cycle. And we have observed that in natural frozen embryo transfer cycle, if progesterone was below 10, the clinical pregnancy rate obtained was significantly lower than the pregnancy rate obtained when progesterone levels in natural cycle were above this cutoff of 10. So this is still an important point for us. And the question is there an optimal level of progesterone? The answer is yes. Only according on the day you are measuring, you should be around 9-10 nanograms of plasmatic progesterone to feel comfortable and to think that the possibility of pregnancy is clear possible. How often this phenomenon is observed in the literature? We have analyzed publications in different authors, and the range of low progesterone level is between 21% and 85%. So it is a common phenomenon. And this phenomenon has clinical importance, and this is why we consider that it's mandatory measuring progesterone before embryo transfer cycle in artificial cycle. Another important point of of the clinical relevance of this artificial cycle is that we have observed in our material comparing the obstetrical outcome, comparing cycles in which there was an ovarian stimulation, that's IVF cycle, or in cycles in which there was an artificial cycle, frozen embryo transfer cycle or oocytonation. We have observed that when we were under an artificial cycle, hypertension disorders were more common. And this has been published extensively in the literature, and this interesting European study show that there was a significant increase in hypertensive disorders and postpartum hemorrhage when an artificial cycle was applied. And you have here the significant difference comparing artificial cycle versus natural cycle, and artificial cycle were stimulated versus stimulated or natural cycle. And the point is that what this author show is that when in an artificial cycle there is an absence of corpus luteum, and if a corpus luteum is not present, there are neoangiogenic changes that are not produced, and this makes the patient more prone to present hypertensive disorders. So this is something that we should bear in mind and to consider this risk from the clinical perspective. Our proposal, just to finalize, is that we should individualize the protocol used for luteal support, and especially in frozen embryo transfer cycle, the protocol is the one I have presented. We measure progesterone. Progesterone is above 10.6. That's okay. We proceed. If not, extra doses of progesterone are given. Measure progesterone after, and if the progesterone is below 10.6 cancel the cycle program. Another cycle with extra doses of progesterone. If the progesterone has been rescued, you can proceed with the embryo. And it's important to give progesterone. It's important to decide when to give this progesterone. And this is interesting French study on the oocyte donation model. What these authors have shown and is the clinical outcome comparing cycles in which no intramuscular progesterone was given, cycles in which intramuscular progesterone was given at the moment of embryo transfer, and cycles in which intramuscular progesterone was given when a pregnancy was conceived, when beta CG has been shown to be positive. And as you can see here, when progesterone was given and the sooner the progesterone was given, the better the results were obtained in terms of increasing live birth rate and reducing the miscarriage rate. And it's important to measure progesterone also on the day when in artificial cycles, when we measure the pregnancy test, and you can see here that the ongoing pregnancy rate was higher when progesterone levels were higher on the day of beta CG determination. And this interesting Danish study analyzed the pregnancy outcome comparing the progesterone values and beta CG values. And as you can see here, when the beta CG and progesterone levels were good in this quartile, most of the live birth cases were here. When, of course, no pregnancy was with progesterone and beta CG was negative, but look here what happened. When beta CG was good, but progesterone was low, most of the miscarriages were in this part of the graph. So you need good beta CG and good progesterone. And here I show you the evolution of our miscarriage rate in cycles in which we apply therapy, artificial cycle, frozen embryo transfer cycle, oocyte donation, and segmented cycles after implantation genetic testing for nucleoides. The figures are quite high, more than 3,000 cycles. We were until 2017 at the miscarriage rate was 13, 16, 23%. When in 2018 we applied this individualized frozen embryo transfer support, we have halved the miscarriage rate. We have significantly reduced our live birth rate thanks to this strategy of measuring progesterone and giving extra doses of progesterone. And to conclude, dear friends and colleagues, we should be convinced that the importance of progesterone in human reproduction. Progesterone supplementation is essential in both in fresh IVF and frozen embryo transfer cycle. You can administer different forms of progesterone by different routes, but you will achieve good results. And the reason is that when progesterone levels are low before embryo transfer is performed, you are at a risk situation for miscarriage rate, high miscarriage rate, and a lower live birth rate. This is a common phenomenon that ranges around 40 to 50%, and the most important factor associated with this low progesterone level is the prior existence of a low progesterone level in the previous cycle. And of course, we need further study, prospective and randomized study showing the value of this strategy of individual. In my opinion, the final measure message of my talk, that you should measure progesterone in each patient, and you should individualize the dose of progesterone according to the specific levels of plasmatic progesterone in each patient. Thank you very much for the talk, and we will be ready for questions at the end of this part of the. Thank you very much.
Xin cảm ơn thông tin bài báo cáo của tiến sĩ Pero Bari. Tiếp nối chương trình là bài báo cáo của tiến sĩ Paul Piet, chuyên gia dược lâm sàng Đại học VUB Bỉ với chủ đề Progesterone, những điều cần biết trong hỗ trợ hoàn thể.
So thank you, Pedro, for this fantastic introduction. And what I want to share with you is my 40 years experience in the development of progesterone formulation for luteal phase support. So my presentation of today will focus on progesterone, what to know about luteal phase support, and it will be a nice complement, I think, comparing to your nice previous presentation. So that's my conflict of interest slide, and with two recent papers on progesterone supporting the presentation of today for those who are interested in having more details about the pharmacodynamics of progesterone. So from general awareness to practical experience, what did we learn from clinical trials? And for sure, we need to balance not only effectiveness, as Pedro was mentioning, but probably also to have a look to safety. So let's start first with the guideline and the recommendation. And as you may know, in 2012, there was a committee opinion published by the American Society for Reproductive Medicine. But you know, we are in Europe. And so looking at the guidelines from the ESHRE development group, they updated their recommendation, and it was released in October 2019. So the questions for luteal phase support are what about dosing, when to start, when to stop, and for how long. So good practice emphasizes a huge variation in the progesterone daily dose, and it was depending the route of administration. And as Pedro was mentioning, for example, you are comparing 600 milligrams progesterone by vaginal oil capsules to 25 milligrams only in subcutaneous route of administration. So interestingly, the dosing of the natural progesterone has evolved empirically. And this makes a full sense because probably, as Pedro was mentioning, depending the procedure, you will need, for example, 400 to 600 milligrams a day micronized progesterone in oil capsules for natural cycles, but probably you will need higher doses, even from 800 to 1,200 milligrams daily for artificial cycle or for stimulated cycle. So depending the procedure, depending the route of administration, the daily dose has to be taken into consideration. And the second question is when to start the luteal phase support, and that was a question from the audience. And the ESHRE expert committee recommends for sure, and we all agree on that, that we have to start the progesterone as soon as possible, the evening of the day of oocyte retrieval, and certainly not later than 3 days after the oocyte retrieval. And this was fully supported by a systematic review by the group of Mohammed published in 2019, showing that how earlier you initiate your luteal phase support after the oocyte retrieval, or better. So the question now is when to start, and starting when for how long, and for the duration of the luteal phase. There's an ongoing debate, but based on the ESHRE recommendation, progesterone should be administered at least until the day of the pregnancy test. However, the NICE, National Institute for Health and Care Excellence, 2013, were saying that the evidence does not support continuing any form of treatment for luteal phase support beyond 8 weeks of gestation. So there is an ongoing debate, even if we know that in clinical practice, most of the IVF centers around will prolong the luteal phase support in after 8 weeks or even 12 weeks of gestation. So, looking now at the progesterone or the tip of luteal phase support, which is optimal, we all know that progesterone has been recommended for a very long time after IVF or ICSI. And it remains in October 2019, the last recommendation by the FDA expert group, progesterone remains the best and probably the strongest recommendation. However, when you look at the recommendations for other progesterone, such as dydrogesterone, the recommendation is conditional. And even in October 2019, they updated their recommendation. And as you can see, they didn't about the safety. And that's really a concern. Dydrogesterone, for example, is an orally active progesterone, which is different in structure from natural progesterone. And that's the reason why the expert committee raised some concern regarding safety for the offspring. And the group of development expert committee consider the database, even of the recent Lotus 1 and Lotus 2 studies to be insufficient to make a firm statement on the rate of congenital anomalies because there was a lack of long-term offspring health studies. And that's probably the key message coming from the ESHRE committee. We need more studies to reassure us about the safety. And regarding efficacy, when comparing dydrogesterone to progesterone, even if the ongoing pregnancy rate were similar, however, in the meta-analysis, the results for frozen and fresh transfer cycles were poor. So there is some question about the efficacy of synthetic progestins or oral progesterone in fed cycles. Progesterone supplementation versus placebo or no treatment improve definitely clinical pregnancy, ongoing pregnancy, to recommend luteal phase support. What I want to emphasize in this Cochrane review is the not negligible rate of ongoing pregnancies in the placebo group. And that's a key and important message. If you look at all the non-studies, comparing one luteal to another, you will need to randomize at least 1,000 patients to make this difference between the placebo effect and the luteal. Non-inferiority study need to be powered with non-inferiority, and that's a key message. Now there is a general awareness about several luteal phase support options in assisted reproduction. And if you look at the most recent updated Cochrane review 2015 published by Van der Wiel, looking at the comparison between intramuscular versus oral progesterone or rectal progesterone, vaginal versus oral rectal, low dose progesterone versus high dose vaginal progesterone, micronized progesterone versus synthetic progesterone, vaginal ring versus vaginal ring, vaginal progesterone versus vaginal progesterone. You know, at the end of the day, the Cochrane review concluded that there is insufficient evidence to implement any one particular protocol. So, what about pharmacokinetic and pharmacodynamic characteristics? The tips of medical phase support were compared. The intramuscular route of administration, and we all know that it will lead to some discomfort for the patient because of painful injection, versus the vaginal progesterone, which probably provides minimal or no discomfort for the patient comparing with the injections. In terms of pharmacokinetic, you can see that administering intramuscular progesterone, that will lead to supraphysiological blood progesterone levels. And that's something that we have to keep in mind, certainly if we monitor our progesterone as a level as recommended Pedro. And for sure, you need a nurse assistant because you treat twice weekly injection. And even in a lot of cases, due to the presence of oil in the injection, even subcutaneous or intramuscular, you can have some allergenic or dry abscess reaction, even acute pneumonia. So there is definitely a different pharmacodynamic profile between intramuscular and the vaginal route of administration, delivering very constant systematic progesterone levels, avoiding the first line hepatic effect, and that's the reason why we vaginal to oral, because probably the vaginal route of administration is the safest route of administration to bring progesterone where it is supposed to work. There is a first uterine pass effect, who was nicely described by a group of miles a long time ago, and irritation or discharges, which can be a problem with the vaginal route of administration, will be highly dependent of the formulation. So what about patient satisfaction? If we are all happy with efficacy, we have to look certainly to the patient satisfaction, and that's the domain where we can make some more progress, because the all those routes of administration are not probably the at least the group of Janupolsky, Fertility Sterility 2010, look at patient satisfaction comparing vaginal route of administration to the muscular injection. And you can see that in terms of convenience of administering, ease of administering and dosing, and overall satisfaction of patients, the odds ratio was in favor of vaginal progesterone. It was statistically significant, odds ratio of about 40, favoring the vaginal route of administration. But what about luteal phase support in clinical practice? Patient satisfaction and so on are in randomized trials. It's not the real life. So what did the group of Weisbusch published in FBO online twice, in 2012 and in 2014, based on a web survey in 82 countries, based on more than 280,000 cycles. As you can see, vaginal progesterone only as a luteal support was increasing across years from 64% to 77% from 2009 until 2018. And surprisingly, you can see that the users of oral progesterone, which is by the way dydrogesterone only, was staying below this rate of 2% across years. And so that is a little bit surprising, knowing the recent publication of Lotus 1 and Lotus studies available for this survey, this updated survey. So what we have to keep in mind that vaginal progesterone is higher concentration than if it was administered by intramuscular injection. So you can see the tissue concentration optimal in the endometrium comparing to despite higher plasma concentration following the intramuscular injection, you have 5 to 7-fold more progesterone comparing to administrative route injection. So that's pharmacokinetic, but what about the clinical trial? And if we look at all the comparative trials between progesterone vaginal capsules and progesterone by intramuscular route administration, overall, the clinical pregnancy, even live birth rate, are similar. And in this study, there was the first study by one of the pioneers in the luteal phase support, my good friend Johann Smith, published in Human Reproduction 1992. This comparative study showing that there were significantly fewer miscarriages in the vaginal group of administration comparing to the intramuscular group, and that was statistically significant. And there was for sure a trend for a higher implantation rate with the vaginal route, but the statistical level was borderline. So we were looking at other randomized controlled trials comparing several formulations for vaginal progesterone. And as you can see in publication by Christina Berg, that is Human Reproduction 2012, that was a randomized multi-center controlled trial in 80 centers in Denmark and in Sweden, more than 2,000 patients, and they compare a vaginal formulation of progesterone gel, you all know gel, comparing to micronized progesterone in oil capsules, well known as Utregestan, and surprisingly, the positive ACG and the clinical pregnancy rate were significantly higher with the vaginal capsule, means here means that it's in favor of the progesterone capsules. And the equivalence of treatments, even in terms of ongoing pregnancy rate, could not be demonstrated, and that's because the 95% confidence interval for the difference in ongoing pregnancy rate, you can see here, in between minus 8.2 to plus 0.1, did not lie clearly within the pre-specified equivalence interval of minus 7 to 7. So is it a question of formulation? Is it a question of dosing? You know that gel is only 90 milligrams a day. Micronized capsule is 600 milligrams a day. Anyway, and based on the previous speaker, you all know that there is a trend, and certainly in fed cycle, to increase the daily dose. And even some patients, as Pedro was mentioning, they need an additional subcutaneous progesterone. So different progesterone genes may differ in their hormonal activity, for sure. But what about safety? And there are plenty of publications looking at the differences in safety comparing natural progesterone to synthetic progesterone genes or other progesterone. It goes about cardiovascular events, venous thromboembolic risk, and even breast cancer risk increase in hormone replacement therapy in postmenopausal women. So a lot of data showing that there is a safety difference in between progesterones. And when we were looking at the impact of oral dydrogesterone during the early pregnancy, what we saw, and that's in the Lancet, that there was a clear association between oral intake of dydrogesterone during the early pregnancy and the congenital heart disease, based on this case control study published in the Lancet. And the odds ratio was even adjusted, was 2.71, highly statistically significant. That was a retrospective case cohort study in the band de Gaza. So for sure, this preliminary study needed some confirmation. And so 2020, the group of Gideon Coren, looking at the cardiac arrest defect induced by maternal medication, published in Prenatal Cardiology Journal 2020, the data also published in the International Model Risk Journal online. They published the data from a huge database from the Maccabi Health Service. And they were looking at the rates of congenital malformations among babies exposed to utero during the first semester of pregnancy. And that's very important, in the luteal phase, dihydrogesterone between 1999 and 2016. So those data were very interesting because Gideon was not looking only at natural pregnancies, but also to IVF. And very recently, all those data were confirmed in a publication last month in Expert Review of Clinical Pharmacology, which is an international review with a very high impact factor, about 4, looking at the fetal safety and the medication that we use to treat infertility. And so what Gideon Coren was looking at is all the previous studies where dihydrogesterone was administered for luteal phase support in assisted reproductive technology, including the 2 recent studies, the Lotus 1 studies, and looking at the odds ratios of selected congenital malformations in the children exposed to dihydrogesterone versus an unexposed control group after excluding those exposed to IVF, or in the patient and the mother pregnant from IVF procedure after adjusting for potential confounders. So the conclusion was that dihydrogesterone conferred teratogenic after exposure to the recommended dose of a pregnant woman. The risk of hyposphagia was statistically significantly increased, and there is some biological plausibility by the known effects on the male genitalia, and the risk for spina bifida also will be proven to be statistically increased in the dihydrogesterone group, and also with a nice biological plausibility proven by the decrease in folic acid levels. So looking at all the results of this publication by Coren, he put the data in a meta-analysis looking at the malformation after dihydrogesterone intake, and even if they were looking at a comparison with fixed effect or random effect, they came to the same conclusion that there is a significant increase. It doesn't cross 1 unit, and there is no heterogeneity in the trials, including meta-analysis. So quite consistent data suggesting some safety concern, as it was mentioned by the ESHRE expert group. So why is vaginal progesterone still considered by most the first option? And also, you know that there was a case report 2020 published by the group of Malherbe, mentioning that there was a case of a delayed fulminant hepatic failure from dihydrogesterone-related in vitro fertilization therapy. And this hepatic failure required liver transplantation, and the baby was lost 2 days before the surgery. In clinical practice, in this publication, the Fertility Center in Singapore, they were looking at the live birth rate and the safety profile of dihydrogesterone for luteal phase support in ART. It's a study with a huge number of patients, and surprisingly, the fetal anomalies were detected in 2.4% of live births. And very interestingly, all the women who terminated their pregnancies did so due to fetal anomalies detected on the fetal anomaly scan. So the women were pregnant after 12 weeks, but they decided to stop their pregnancy for those fetal anomalies. If you look at the clinical trials that we have for the time being, and you all know the Lotus 1 and the Lotus 2 studies, and the authors of the study claim that investigating the long-term safety of progestogen formulation for lethal should be an important area of future research. So the Lotus 1 and the Lotus study didn't fix the safety issue, and that was fully endorsed by most of the European regulatory agencies, claiming, based on the precautionary, that safety is the first concern in pregnancy before efficacy. And that's the reason why, for example, in Switzerland, they consider that dihydrogesterone is contraindicated as long as we have no more safety data. And even in France, the ANSM statement is very strong. This dihydrogesterone product is not indicated in pregnancy. Looking at a comparison with several formulations of what about the efficacy of several progesterone? Is it a question of dosing, or is it a question of the route of administration? So that was an interesting publication, a presentation by the group of Lasserre in 2017 at the ESHRE meeting, comparing the efficacy of vaginal progesterone as standalone product versus subcutaneous progesterone as standalone product. And as you can see, implantation rates, biochemical losses, were all in favor of the vaginal progesterone. But the question is, was it related to the route of administration, subcutaneous versus vaginal, or was it related to the daily dosing, as Pedro was mentioning? It's so important to have enough progesterone to support the luteal phase. And as you can see, we are comparing apples and pears. 200 milligrams twice a day, 600 milligrams 25 milligrams a day. So pharmacokinetics and pharmacodynamics are crucial to try to explain how works luteal phase support in IVF. But at the end of the day, looking at the public assessment report edited by the MHRA for the European approval of the subcutaneous preparation, there was a clear suggestion by the MHRA that subcutaneous progesterone are not as efficacious as standalone, comparing to vaginal preparation, taking into consideration the proposed indication and the overall uncertainty regarding the optimal dose, the timing of the use of progesterone for luteal phase support. So they were saying that it might be an option for a woman. So based on the European assessment report, definitely subcutaneous progesterone is a second-line therapy. And as Pedro was mentioning nicely, it can be a very interesting route of administration to add some more progesterone when the progesterone blood levels are not sufficient. So if you look now at the efficacy in the two pivotal studies with dihydrogesterone and oral progesterone for luteal phase support, again, it's interesting to mention that the authors consider that the study was not powered for live birth comparison, and that probably live birth rate may be of greater clinical interest for sure. So the conclusions from both Lotus 1 and Lotus 2 studies were saying that the differences between treatments in terms of live birth rate in those two studies should be therefore viewed with caution. So even if we have nice efficacy endpoint, those endpoints were related to the ongoing pregnancy rate, and the study was not powered for live birth. As mentioned in my previous slide by the Cochrane review, it's important to power your study to demonstrate non-inferiority on live birth rate, and for that you need more than 12 patients in a non-inferiority study. Now, is it worth to monitor our progesterone? And if I understood the presentation by Pedro, it's really crucial in FET cycle. So how you can monitor progesterone levels? If you administrate vaginal micronized progesterone by oil capsule, you will have a first uterine pass effect with very high endometrial progesterone concentration, as I mentioned, and there is a physiological diffusion of progesterone, leading to a progesterone steady-state plasma level of about 12 nanogram per ml when you administrate progesterone by vaginal capsule at the daily dose of 600 milligrams. And as Pedro was mentioning nicely, in some patients, it's not the case because there is a huge variability. And so it's mandatory to add some progesterone, and you can do by continuous administration, as Pedro was suggesting, or even by oral progesterone. But what about the monitoring with dydrogesterone? You have a nice oral intake, but there is an intensive hepatic first pass metabolism in 20 alpha dihydrogesterone, which is absent in natural pregnancy. So you need a highly specific and chromatographic assay to monitor biological levels of dihydrogesterone or 20 alpha dihydrogesterone active metabolite. And there is no validated normal value for those progesterone. And in a very old study by the group of Balache in France, published in Fertility Sterility 1980, comparing the plasma progesterone levels during the luteal phase of a normal cycle, comparing the levels during treatment in the same woman with dihydrogesterone, you can see that you don't have any difference in progesterone level. So you cannot monitor progesterone when you administrate oral dihydrogesterone. I think that you saw this slide presented by Pedro, and so that's true that progesterone is an essential hormonal marker in the luteal phase, certainly in FET cycle, and this set-off level of about 10 nanogram per ml was replicated in several studies. Also, the group of Cédrin-Durnérin was suggesting a very nice predictive model of live birth rate by serum progesterone levels on the day of embryo transfer. And as you can see, confirmed by this significant drop curve, there is a clear linear correlation to higher your progesterone levels the day of the embryo transfer, to higher your biochemical pregnancy, ongoing pregnancy, and also live birth rate. But the significant difference were significant for clinical pregnancy, however, it was not significant in biochemical pregnancy or in live birth. So this study needs to be replicated to be sure that we can find an optimal cutoff level, but until now, all the study, as Pedro was mentioning, is about 10 nanogram per ml, which is in line, in fact, the old definition by Jordan on luteal phase deficiency, saying that below the 10 nanogram per ml. However, if you look at this publication, very interesting, by the group of NRC, published in Reproductive Medicine Biology 2018, they were looking at the effectiveness of higher dose of vaginal progesterone. In this study, they administered 1,200 milligrams of vaginal progesterone capsules in oil, and they compared to a lower dose, 900 milligrams, and they had a higher clinical pregnancy rate with the high dose transvaginal progesterone comparing with the lower dose, despite a blood level which was not more elevated comparing to the 900 milligrams. So again, we have to be very cautious about the concentration of progesterone level. We need an accurate lab. We need to measure progesterone at the same time in the day, probably better in the morning. And we have to take into consideration age and weight and all the determining factors. So, in conclusion, why should vaginal progesterone be the treatment of choice for fertility support? And that's the case in Finland 30 years, and it's really confirmed even by the guidance by the expert group. We prefer vaginal progesterone because we have this first uterine passive delivering high local uterine concentration with targeted action. That's the only route of administration where you bring immediately progesterone where it's needed. For sure, vaginal progesterone, as Claire Bourguin was showing nicely in 1990, it delivers optimal endometrial transformation based on biopsy, and most of the endometrium are in phase, which is mandatory for an optimal luteal phase support. At the pharmacokinetic point of view, the bioavailability is optimal because we have very stable plasma level comparing with the intramuscular route or the subcutaneous route of administration, delivering peaks and valley, which is not good for the endometrial receptivity. And for sure, with vaginal progesterone, you will have higher plasma level at the steady state comparing to the oral. And that's why we prefer to use vaginal route for luteal phase support. It's convenient and user-friendly because the ease of administration, as Jan Polski was showing, providing a scope of self-administration. It has been extensively used, not only in fresh, but also in frozen cycle. It's supported by clinical evidence with the most important clinical endpoint, live birth rates. And last but not least, you can avoid the first liver gas metabolism using vaginal instead of oral progesterone because you will have no side effects or irritation at the gastrointestinal tract. So I thank you for your attention. Ready to answer your questions.
Dạ vâng, xin cảm ơn phần chia sẻ của tiến sĩ Pau Piet. Tiếp nối chương trình là bài báo cáo của Phó Giáo sư Sandro Gili, chuyên gia sản phụ khoa Đại học Perugia, Ý, với chủ đề có thể chuẩn hóa trong chuyển phôi IVF. Xin kính mời quý bác sĩ lắng nghe.
I think you see my slide, my first slide, and I want to be a little bit more practical, I would say, because when we talk about the endocrinological point of view, we talk some very important issues, but we go to the practical point, to the technique of the transfer. You see, this is the last report showing that Italian National Italian Registry, the last report where you say that there is no significant increase in the pregnancy rate during the last years. So we did not anything better probably, but where can we will change something in increasing our life to something in ovarian stimulation, in agriculture, from the biological point of view. We can do, again, we just said, we just heard about the PGT analysis. We heard about the endometrial receptivity and importance of progesterone. We said about this, but we can do something with probably with the embryo transfer or the number of embryos. Although we know the scientific, the recent scientific report say that there is no big difference in the number of embryos. Increasing the number of embryos probably will increase the multiple pregnancy, but not our implantation rate. Nothing we can do with the ovarian reserve of the age of woman. Probably plenty of things we can do with the male factor. So these are the topic, the variables where we can act to probably increase our life birth rate. But at the end of the day, we can do something to modify the embryo quality. We can do something with the improving the endometrial receptivity. We heard with the importance of progesterone, major progesterone at the time of embryo transfer. We can do also something during the last act of our procedure, transfer technique. Remember that this procedure, this step of the procedure is very important. We can do the best ovarian stimulation, we can provide the best embryo, and we can manage wonderful endometrial receptivity. But if we fail to do a transfer technique, we fail all the procedure. And we have to remember this is a operator dependent variable, and therefore it's important probably to effectively standardize. These are the variables where we can manage to something with ameliorate or optimize the embryo transfer technique. We have to remember the uterine anatomy and physiology, so the position, the geometry of the uterus, the presence of the contraction. Maybe we can do limit the presence of contraction. We have to remember the importance of the operator, and in the next slide we will see something about this. The technical procedure, the insertion of the catheter, the location of the tip of the catheter, the speed injection of the fluid, the extraction when we withdraw the catheter from the cervical canal, from the uterine cavity, and the presence of the ultrasound guidance. The type of the catheter is also important, and also the tip, how is realized the catheter in its diameter, and also the catheter. So these are more or less important variables where we can manage to improve our technique. And this very recent paper, the importance of the operator was discussed. As you see from the left of the slides to from the top of the slide until the bottom of the right part of the slide, you see that different operator with different moderation for the pregnancy rate. So it means, for example, here we have the worst operator in the reduction of the moderation, and here in this part of the slide you see the best operator with an increase in the moderation. Now, you can speculate that probably it is important the experience of the operator. Well, if we pay attention to this, operator number 1 and 2, for example, the number of procedure was 270 and something like 3,000 procedures. So high number, very high number for the operator number 2. And if you see the ongoing pregnancy rate, in this case adjusted, you see that the operator number 2 has something like 22% versus the 27% of operator number 1. So the number of procedures doesn't seem to exert any important influence on the ongoing pregnancy rate. And in fact, you see that with the logistic model, they applied. In this case, they showed that the importance is the femal age for the, obviously, for the pregnancy rate, the femal age, the number of sites we retrieved, the number of embryos transferred, and also the fertilization rate, but not the operator experience. So this is that doesn't seem to represent an important variable for the our results. A couple of issues that are where we can meet really definite argument. The importance of the transabdominal ultrasound guided embryo transfer, the possibility therefore to follow the catheter, the outer catheter and the inner catheter inside the uterine cavity. It is something that we all agree that is very important and can improve our procedure. There are a couple of publications demonstrated, this one 2 years ago, demonstrated that if we compare the transabdominal ultrasound versus the embryo transfer with only the clinical touch, the clinical pregnancy rate and the live birth rate were significantly higher in the case of ultrasound embryo transfer followed by ultrasound. So the importance to follow the catheter inside the cavity and to see exactly where the tip of the catheter is located. Also, this Cochrane of 4 years ago demonstrated the same thing for live birth and for ongoing pregnancy rate. Ultrasound guided embryo transfer is superior in terms of results to the clinical touch. We all use, I believe, the soft catheters. These are the fluoroscopic image of some trauma determined by the firm catheter, which are, I hope, completely abandoned. At the same time, we have to remember the importance to remove the excess of mucus and debris from the cervical canal before introducing our catheter. Many publications, this is one of them, and the clinical pregnancy rate, implantation rate were significantly high, together with the live birth rate in case of removal of this mucus. The speed of injection is the that we can have a different injection during the injection of the fluid and the embryo. So we can do a different way, and we can analyze also different results depending on the speed of injection. There is a statement of the ASRM of 2017 saying that there is insufficient evidence to recommend any injection speed of the catheter at the time of embryo transfer, but there are some experimental model, and this was very, very interesting in this case. This little table in which the uterus was stimulated with the uterine cavity and also with the cervical canal and the cervix. So they tried to simulate exactly what happens with slow injection or fast injection inside the uterus, even trying to simulate the angle of inclination and anteversion or retroversion of the uterus. And if we have this simulation, this is the simulation of the uterine cavity, you see that with the low speed of injection, with a 30 degrees angle of inclination of anteversion of the uterus, you see that the blue ticket, that means the fluid that we are injecting, remain very close to the fundus of the uterus, and the tip of the catheter is below. But this is not happening during the high speed of injection. The fluid remains very close to the tip of the catheter and does not remain exactly where we probably would like to be very close to. And at the same time, the difference in the speed of injection is important also for the development of the embryos. This is an experiment of on mouse embryos. with a fast injection speed of more than 1 meter per second and slow injection. And the development rate was the slowest in embryos exposed to the fast injection, together with the elevation and apoptosis index, which was 17.6 versus 5.6 in the slow injection. So it means that we have a biological demonstration and physical demonstration that the slow injection of the fluid could be very important for our results. Another issue very important is where to locate our tip of the catheter. How many millimeters from the fundus we can measure this, for example, even if we do the embryo transfer with ultrasound guidance. In this study, they divided 4 groups of patients in which the tip of the catheter was located at less than 5 millimeters from the fundus, between 5 and 10, 10 and 15, and more than 15 millimeters. So very close to internal cellular loss. And if you do like something like this, you see that your pregnancy rate and your implantation rate decrease significantly. So we have worse results with the tip of the catheter more than 150 millimeters from the fundus. But we have to consider also that if we follow our fluid and the hypercoic area immediately after the transfer represent the embryo flash position, we follow this position 60 minutes after the embryo transfer, we can assess the movement, the migration of this hyperechoic area. So therefore, we could have the migration immediately after the transfer to the fundus. This is one possibility, and this can be seen 1 hour again after the transfer. But also, we can assist to the migration to the cervix. Well, the fundal migration group, the cervical migration group, or the static group where we don't have any migration, were divided in 3 groups, this patient, and were observed the pregnancy rate and implantation rate. As you can see, the cervical migration group demonstrated a lower, significantly lower implantation rate and pregnancy rate. So we have to do something to improve our pregnancy rate and do something to avoid the cervical migration and to render possible as much as possible the fundal migration. Another study of 2 years ago in which the same thing was demonstrated, in which cervical migration demonstrated a lower, significantly lower pregnancy rate. Also, the quantiverbal analysis demonstrated that the age obviously was important, but also the embryo flash migration toward the fundus was able to increase our pregnancy rate. And regarding this migration, is important also this very recent which fresh and frozen transfer were compared, and in this study they demonstrated that the cervical migration was more frequent, significantly more frequent in fresh cycle compared with frozen cycle. You have to remember that in this study they did the fresh cycle in day 3 and frozen cycle in day 5. They probably justified this migration versus the cervical canal in fresh cycle, probably with the different hormonal milieu present in the uterine cavity and in myometrium. Probably the presence of lower estrogen level and lower contractility in frozen cycle is able to induce a lower percentage of patients with the cervical migration. And if we have cervical migration, again, they get a lower pregnancy rate again. So the same demonstration that in this case is important to avoid this possibility. What about the time interval between the embryo is loaded and the fluid discharge inside the uterine cavity? Important to determine probably the time interval. Let's see the study, and they again subdivided the population in which the time interval was less than 30 seconds, or between 30 and 60, and between 60 and 2 minutes, and over 2 minutes. So in this case, you see that probably percent of non-easy transfer obviously were mostly grouped in the 4 groups. So in the fourth group, which means that probably when we have a delay in our transfer, probably do non-easy transfer, probably because of the antiversion of the uterus, of the stenosis, and so on. But it's interesting in the study that the pregnancy rate and implantation rate, not including, so excluding the non-easy transfers, demonstrated a lower pregnancy rate in cases of the patients in which the transfer there was a time interval more than 120 minutes. So it means that we have to do the transfer in a hurry. I mean, not to go in more than 2 minutes between loading the catheter and discharging the fluid. Withdrawal of the catheter immediately after we have released the fluid and the embryos inside the cavity. It is important to retrieve immediately or to wait at least 30 seconds. We can withdraw immediately. There is no difference absolutely between withdraw immediately and to wait 30 seconds. So we can release the fluid, and then we can withdraw our catheter without any fear. This is one fear. It's important when we hear from the lab that the embryos are still here in the catheter. Don't worry, because there are many studies. Is the one of the study demonstrating that even if we retransfer embryos which were retained inside the catheter, we don't have any problem and any influence on our pregnancy rate. So these are the results in terms of larger rate for people, and p-value demonstrating that for the 2 groups in which there was the embryo retained or no embryo retained, there is no difference in the outcome of our procedure. Should the patient lie down for a few minutes after the embryo transfer? We can answer showing something about anatomy. This is the patient lying down, and we have to remember lying down. The patient has a bladder. The uterus can go down in something like, even if anteversion go to retroversion, or in the thin plane of the vagina. And if the patient is standing up, and the bladder is empty, the uterus then goes to the anteversion, and this is the best position to maintain the embryos and the fluid inside the uterine cavity. So this is the reason why there is no justification to maintain the patient lying down even for a few minutes. And the demonstration of this is published years ago, demonstrating that deliveries were significantly higher in patients with no rest compared to the group of patients in which the rest was done after the embryo transfer. So immediately after the transfer, put the patient immediately standing up, empty the bladder, and by this way you can obtain better results. Even for our little study, for some little group of patients divided by age, you see that we didn't have any difference in the results. But there is a convenience for the logistical point of view, because the patients can leave the clinic as soon as she finishes with the transfer. So, and again, last year was published again another research in which the immediate mobilization after the embryo transfer was demonstrated not to have any influence over the success rate of our procedure. Difficult transfer, sometimes we have the possibility, and this is evaluated around 5–8% of patients, and this is due probably to anatomical position of the uterus, of the angle between the cervical canal and the uterine cavity, or cervical stenosis. And these are the method we can apply to overcome this problem. So the use of the outer step of the catheter, the use of the stylet, or the use of the baculum. You see that even use one of these, your pregnancy rate can decrease significantly. And so be aware about this possibility and try to do your best to do without using these maneuvers. Another possibility is the cervical dilation. If you want to do the cervical dilation before the embryo transfer, remember that it's better to do 1.5 or 2 months before the embryo transfer. This study was demonstrated that the pregnancy rate seems to increase if your cervical dilation is performed 1.5 or 2 months before the embryo transfer. If you have problem impossible to overcome, another possibility, and you know that there is a possibility to do this, the method of transmyometrial embryo transfer, in which we have results that are not completely comparable to the results of the regular embryo transfer, but is the only possibility in some cases, a few cases in which you cannot perform your regular embryo transfer. So this step of our procedure is important, really, and you can fail everything after your ovarian stimulation, after the embryos and so on. But if you fail this transfer with this step of the procedure, you fail. And it's important. This procedure, this step, is performed by personal train, but we have seen that the experience is not influenced. Just to summarize, ultrasound guidance is important. You remove your cervical mucus. You can do direct transfer with the 2 catheters in and an outer, but you can also do the after loop transfer. Use the soft catheters. Tip of the catheters no more than 15 millimeters from the fluus. Low speed injection. Short intratransfer time from loading the catheter to withdrawal. Check for retained embryos, and no bed rest, but stand up after your transfer. Thank you very much for your attention.
Xin cảm ơn thông tin bài báo cáo của Phó Giáo sư Sandrozeri. Tiếp nối chương trình là bài báo cáo của Giáo sư Umberto Simeoni, cựu Chủ tịch Hiệp hội Chu sinh châu Âu, Đại học Lausanne, Thụy Sĩ, với chủ đề hỗ trợ sinh sản có ảnh hưởng đến gen và kết cục thai kỳ. Xin kính mời quý vị bác sĩ lắng nghe.
Thank you very much for introducing me, and thank you, Professor Miranda, for your invitation and for having included a pediatrician in the faculty. Indeed, we are all aware of the current knowledge of the general outcome in children born after IVF. The most important point I express is that most IVF conceived infants and children are healthy, including today at adulthood. However, a number of studies, observational, have shown that IVF is associated with congenital abnormalities, with parental imprinting associated disorders such as the silver rustle or the Wiedemann-Beckwith or Prader-Willi syndrome. Of course, some adverse perinatal outcomes, which have already been discussed, is important as well for the baby, is the birth weight and the risk of preterm birth. And later, an increased risk for neurodevelopmental disorders, including autism spectrum disorders, and an increased risk on the long term at adulthood for chronic disease, especially cardiovascular and metabolic disease. What can be understood from these studies is that certainly for the initial studies, multiple gestations in IVF were a main driver for these outcomes. However, IVF pregnancies have been shown to be at increased risk, also lower risk as well. Then concern has been raised on the role of hormonal stimulation, gamete manipulation, embryo culture and preservation, and potential alteration of the epigenetics, the normal epigenetic imprinting of gametes and of the embryo. Anyway, we have to be aware of the potential biases and additional risk factors which can be at play, especially maternal as well as paternal health, which influence, of course, the baby outcome and can also influence their own fertility and the causes, especially the lifestyle and environmental causes of infertility are certainly important biases in the studies we have to understand which is the outcome, the long-term outcome, maybe for IVF. For example, arterial hypertension during adolescence has been shown to be more frequent in adolescents born after IVF. It's a study performed in Italy. And these patients indeed show, including after 5-year follow-up up to adolescent starting in childhood, that 3 biomarkers of cardiovascular risk are altered in those who were conceived with ART. And it is flow-mediated dilation here on the left, a pulse wave velocity, which is a measurement of period in the intima media thickness, which are classical risk factors for later cardiovascular disease. And indeed, these adolescents have a higher systolic and diastolic blood pressure at young adulthood, which is, although the difference may be considered small, but having such an increased blood pressure at adolescence is a significant, very significant risk factor for maturity, for the age of maturity, developing, for example, stroke or coronary heart disease. Anyway, we have to keep in mind that the control subjects are not the only difference between the ART and the control subject is not only ART, and the causes again of infertility in the parents, which might either persist or be inherited due to epigenetic mechanisms in the offspring, can be at work. And so we have a problem of defining the controls when we evaluate the impact of ART. Other point is that indeed fertilization and the reproduction occur at a key time period of life, which is early. And this is a period of development. And we know today that the development of a child is mainly driven by the environment. Environment understood as lifestyle, behavior of parents, including before conception, and of course also environmental toxicants or stress simply. We know that what happens at the beginning of life shapes the trajectories of health, the life, the risk for later chronic diseases at adulthood, including infertility. The stimuli which can influence these trajectories are very subtle. They can consist in variations within the normal range in terms of nutrition or stress. And you may remind the first studies of David Barker and his group, who showed that the lower the birth rate, the higher the risk of chronic disease in late adulthood. But they studied mainly people with a birth range. So very subtle changes early in life can have significant consequences. When it comes to IVF, and especially when multiple embryos were transferred, and we are a follow-up up to adulthood, referring to these time period complications of multiple pregnancies, certainly can influence the outcomes of these babies and be a major factors, including preterm birth or intrauterine growth restriction, or the exposure to maternal diabetes or overweight and other factors. So is ART, which is the role, the specific role of ART remains really to be defined. We know that this period of development is under the influence of epigenetic changes, which just translate the effects of the environment on the genome. And this is also at work in the cause of infertility itself. We observe another pandemic, which is the decrease of fertility in the world, including in Africa, is anticipated in the next decade and is already a reality today, and is very likely to have, again, environmental causes. The environment considered as a general container, including behavior, lifestyle, and of course, passive or active exposures to toxicants and to stress. Also, of course, we are just dealing with IRT at a period of normal changes in the epigenetic imprinting of the offspring. And this is due to the development of the primordial germ cells, which undergo a complete erasure of their epigenetic imprinting, more or less completely. They, as some acquired traits of the parents, can be inherited, and they can so contribute to not the genetic but the epigenetic characteristic of the offspring. So big changes in epigenetic imprinting exist there, and also the development again is an epigenetic process per se, because epigenetics, as you certainly know, alter gene functioning, but not gene sequence. They set up the regulations of gene functioning or on the long term. So it is a normal process which can be altered in the presence of an altered environment. And when an individual develops, it can be compared to what happens in a stem cell. A stem cell commits itself to differentiate into a neuron, for example, by silencing part of the genome. Each cell contains the whole genome, and overexpressing another part of the genome, which is necessary to function as a neuron. This is an epigenetic natural mechanism. So development is per nature epigenetic. So that could be why among the potential effects of IVF on the offspring, there is one key point which seems quite robust, which is an increased incidence of congenital defects related to parental imprinting disorders. These syndromes we all know, Angelman, Silver Russell, for example, are not strictly of genetic nature, but of epigenetic nature. They depend on the equilibrium between the allele of the gene or from the mother. And this difference is driven by epigenetic mechanisms. And one point which is relatively clear is that the incidence of disorders is higher in patients born after ART compared to spontaneous pregnancy in this study from the group of Atari in Japan, with an incidence of the Silver-Russell syndrome, which is nearly tenfold observed in spontaneous pregnancy. So if we take into account the genetic mechanisms, we have to be aware that they have an extremely potent impact. Indeed, we certainly know that it's about methylation or histone modification or non-coding RNAs, which are generated according to the signals received by the early environment. And an example of the importance of this phenomenon in development, in child development, is in bees colonies. You may know that in a colony of bees, only the queen is able to reproduce, and this is not genetic, it's not chromosomal, it's simply due to early nutrition, which means early environment. The queen receives a very rich nutrition for a longer time than the other bees, the royal jelly, and this is, as a consequence, she can live longer, she's bigger, and she is fertile, which the other bees are not. So epigenetics have importance, and they constitute an epigenome, which is as important of the genome, maybe even more, to determine the fate of an individual. But the epigenome, contrary to the genome, is flexible. It's not structural. The genetic imprinting, either methylation or histone modification of non-coding RNAs, are dynamic and reversible, although they can stay over the life course and even be inherited for a few generations. And they translate the impact the early environment, and so they control developmental plasticity and allow one single genotype to develop into an infinity of phenotypes. And in turn, these phenotypes will be as well behavioral and more or less sensitive to the environment. So the epigenome introduces a much larger biologic diversity in the genome, our 20,000 or 30,000 genes which are at work. They are tuned by the epigenetic machinery for life during early life, depending on the early environment. And this has been very nicely shown by Conrad Waddington in the 50s, just at the time when the double helix structure was found by Crick and Watson of the DNA. And it showed very nicely, while we knew really little about genes and the epigenome at this time, he coined the term epigenetics and understood that the trajectories of development or of life are influenced by the environment. If this is an individual or a cell, it can run according to several directions early during development, according to the forks and the channels it can encounter. Amazingly, Waddington was the supervisor of the thesis of Dr. Edwards, who was let's say, mother of the first in vitro fertilization and the baby Louise Brown. So the result is that we have these trajectories of life, which can be, in terms of health, either favorable in green or less favorable in blue or in red, depending, of course, also on the major adversities encountered by the individuals. But the slope of these trajectories is determined very early by the epigenome. Both constitute the position and the developmental plasticity, which is major at the beginning of life. So this is important because if we intervene late, it's much more difficult to correct this than if we can intervene early and provide an early prevention for health. Finally, there is a transgenerational inheritability of early epigenetic imprinting from one generation to the other. So this is important because it stresses the point that starting prevention or interventions in the preconception period is key to be successful. As Roland de Vlieger showed it is very difficult to intervene, very much time-consuming, and certainly it's not simply by saying to the people, well, you wish to have a baby, you should eat better, have food, and have an increased physical activity, and that's it. People need to be coached, to be supported over time, and it's certainly an important limiting factor to our intervention. This shows also that the current pandemics we observe in chronic diseases such as obesity are likely to be leveraged by this transgenerational health built into the effect of the reproductive cycle and the amplifying effect of this cycle. I will end with simply reminding that anyway, being born naturally or after ART is a major risk factor for later health. Here are the differences in an important meta-analysis of blood pressure is a historical diastolic at young adulthood in low or very low birth weight infants, and there is a clear difference of several millimeters mercury at young adulthood, major in women. This is independent of the low birth weight, except maybe that having a preeclampsia, it may even increase this effect. But it is an early important risk factor for later health. And in these babies, for example, we observe that there is an alteration of the endothelial angiogenic capacity. As shown here, for example, in how endothelial progenitor cells from low birth weight infants on the left part of the picture build a new vessel compared to control term newborns endothelial progenitor cells. Very nice vessels compared to the reduced capacity of low birth weight EPCs infants. These changes are related with a phenomenon which is quite frequent in altered developmental programming, which is accelerated cell senescence due to stress, to sirtuin-1 deficit, itself related to changes in the histone 3 methylation, which is an epigenetic mark of low birth weight. So this is less the case today, as only one embryo is often transferred to the mothers. And also, we have a very interesting study which was able to perform an epigenomic-wide association study between patients, adult patients born after ART or born for control subjects, healthy subjects, and they were able to have also neonatal blood samples from these patients. And they found that while there are changes in red here in a number of genes, of the methylation genes, differentially methylated regions, these are the red lines. The blue lines represent the same genes, the methylation of the same gene at adulthood. So a number of changes which are found in the neonatal period of genetic changes do not persist in adults. We just have to keep in mind that these adults were healthy subjects, but this is, I would say, a bit reassuring. I will conclude because we have little time. Early exposure to environmental variations of stress, even if they are subtle, lasting long, silent effects on all components of health, physical, mental, the preconceptional period seems important as it is sensitive to epigenetic reprogramming. Epigenetic programming is a normal phenomenon. Developmental programming is normal. But if the cues received from the environment are, and of course, gametes and embryos manipulation and culture have the potential for such changes, possible long-term consequences. But we should keep in mind that anyway, most children are doing well. There is evidence that a range of adverse early life outcomes, including rare parental imprinting related syndrome, are observed and maybe certainly are a problem to be addressed. But the association of ART with chronic diseases at adulthood is also well established by the causality. The association is very complex to be ascertained due to a number of confounding factors at the moment. So finally, also some variations, some genetic is at birth, we need a it persists at adult, very difficult in young. Thank you for your attention.
Kính thưa quý vị bác sĩ, phần trình bày của giáo sư Ampeto Simeonin đã kết thúc bài báo cáo khoa học trong chương trình ngày hôm nay. Sau đây là thời gian dành cho phần thảo luận. Quý vị bác sĩ có thể đặt câu hỏi cho chủ tọa tại Việt Nam là anh hùng lao động, nhà giáo nhân dân, giáo sư, tiến sĩ, bác sĩ Nguyễn Viết Tiến thông qua hai hình thức. Cách thứ nhất là click biểu tượng raise hand để ban tổ chức có thể bật mic cho bác sĩ hỏi trực tiếp báo cáo viên. Cách thứ hai là click biểu tượng Quick and A và để lại câu hỏi để báo cáo viên trả lời. Kính thưa giáo sư, tiến sĩ, bác sĩ Nguyễn Viết Tiến, như vậy sau phần trình bày hết sức bổ ích của báo cáo viên nước ngoài cập nhật những cái vấn đề hiện nay trong hỗ trợ sinh sản, thì cũng xin giáo sư cho ý kiến tổng kết cũng như ý kiến tham luận ạ.
Vâng, trước hết là rất cảm ơn báo cáo viên đã có phần trình bày rất là hay, và thậm chí có một số các quan điểm là cũng khác ngược với những cái quan điểm chúng ta có ở trước đây. Lại tôi cũng xin điểm qua những cái ý chính của nội dung của 4 bài báo cáo, và đặc biệt là nhấn mạnh những cái vấn đề mà chúng ta có lẽ là từ trước đến nay chúng ta chưa làm hoặc đã làm thì làm rất ít. Đấy là về cái vai trò của progesterone hết sức quan trọng rồi. Thì bao giờ khi chúng ta chuyển phôi thì cũng sử dụng, kể cả phôi tươi cũng như phôi trữ thì đều là sử dụng progesterone. Và nhưng chúng ta cũng thường ấy thì không quan tâm nhiều đến cái nồng độ progesterone trong máu trước khi mình chuyển phôi. Nhưng báo cáo của tiến sĩ Pedro Paris, người Tây Ban Nha đấy, thì có nói rằng trước khi chuyển phôi chúng ta nên định lượng progesterone. Nếu theo nghiên cứu thì nếu như nồng độ progesterone mà dưới 9,64 thì gần như là chuyển phôi thì sẽ thất bại, mà nồng độ progesterone nó phải trên 9,64, và có nghiên cứu nữa thì người ta lấy là trên 10 phẩy. Do có lẽ để cho dễ nhớ chúng ta nên lấy ở cái mốc là ít nhất là khi chuyển phôi là nồng độ progesterone phải trên 10 nanogram trên ml. Còn đặt ra tình huống nếu như mà khi định lượng progesterone trong máu mà dưới 10 nanogram trên ml ấy, thì chúng ta có thể tiếp tục duy trì nồng độ progesterone tiếp, và sau đấy chúng ta định lượng lại progesterone. Nếu như nồng độ progesterone mà trên 10 nanogram trên ml thì chúng ta cho chuyển phôi, còn nếu như dưới thì chúng ta hủy chu kỳ. Nhưng lúc nãy thì tôi cũng hơi thắc mắc một chút là nếu như chúng ta cứ tiếp tục dùng progesterone mãi như thế thì dùng đến bao nhiêu ngày, dùng đến bao giờ, hay chúng ta tiếp tục dùng 1 tuần hay 2 tuần để cho nồng độ progesterone mà nó phải trên ngưỡng của nó là phải trên 10 nanogram trên ml. Và chúng ta cũng biết rằng là nhiều khi mà vẫn cứ tiếp tục dùng như thế thì bệnh nhân lại ra kinh. Đấy là một câu. Thế còn tiến sĩ thì cũng có trình bày một thực nghiệm, nhưng cái này tôi cũng không hỏi, chúng ta cũng không hỏi được, nhưng mà cũng là khó, là đối với những cái trường hợp mà khi mà bệnh nhân có thai, mà khi mà thai nghén đấy mà dưới 7 tuần thì người ta cắt bỏ cái hoàng thể thì thai bị chết. Còn nếu như tuổi thai mà từ 8 tuần trở lên mà có cắt bỏ hoàng thể thì thai vẫn tiếp tục phát triển bình thường. Còn nếu 7 tuần trở xuống mà cắt bỏ hoàng thể, sau đấy người ta bổ sung ngay progesterone thì thai vẫn phát triển bình thường. Thì tôi cho rằng nếu thực nghiệm này chỉ thực nghiệm trên thỏ và trực và thực nghiệm trên chuột mà thôi, chứ còn không ai có đạo đức mà thực hiện trên người. Và chúng ta cũng đã biết rồi là về cái thai nghén của người á, thì khi mà thai hết 12 tuần thì lúc đấy mới hết vai trò của hoàng thể. Tất nhiên là nếu chúng ta bổ sung cái progesterone phải bổ sung cho đến hết 12 tuần của tuổi thai. Chứ còn nếu như trước 12 tuần tuổi thai mà chúng ta phá hoàng thể đi thì thai bị sảy, thai sẽ bị chết. Và chúng ta còn có một quy định thành phác đồ, thành chỉ định, nếu những trường hợp nào bệnh nhân mang thai phát hiện có u buồng trứng mà chúng ta muốn cắt cái u buồng trứng đấy thì phải chờ cho đến lúc thai quá 12 tuần, chứ còn không chúng ta phạm vào chúng ta cắt cái hoàng thể thì cái thai đấy nó bị sảy. Thì đối với là trường hợp là như vậy. Còn ông có đưa ra là cái tỷ lệ chuyển phôi trữ, phôi đông lạnh ấy, thì ngày càng nhiều hơn, mỗi năm tăng 20%, và ngay ở Tây Ban Nha thì cái tỷ lệ chuyển phôi trữ khoảng hơn một nửa. Nhưng mà riêng ở Việt Nam chúng ta tôi tính có lẽ chúng ta chuyển phôi trữ trên 80-90%, nên chúng ta chuyển phôi trữ thì nhiều hơn. Thế còn cái khi mà bổ sung cái nồng độ progesterone thì có mấy cái điểm chú ý là thường những người tuổi cao là một, người béo phì là hai, thì chúng ta thường thì trong máu của họ nồng độ progesterone thường là thấp hơn. Và progesterone thì cũng khẳng định là dùng càng sớm càng tốt. Tất nhiên là có người thì cho rằng ngay sau khi lấy noãn thì dùng progesterone trong ngày hôm đấy luôn, mà đấy thì cái nhấn mạnh là dùng vào đêm cái ngày mà lấy noãn. Thế còn có quan điểm thì có thể dùng sau khi chuyển phôi 3 ngày, nhưng mà ở Việt Nam chúng ta vẫn quan điểm là nếu như chuyển phôi tươi chúng ta dùng sớm. Đấy là cái nội dung của bài báo cáo đầu tiên. Thì tất nhiên là về cái có nhiều nghiên cứu, nhưng tôi nốt lại thì cái ngày chuyển phôi thì phải trên 10,64 nanogam trên ml, nhưng chúng ta phải nhớ là trên 10 là được. Thì tất nhiên có một nghiên cứu nữa là trên 9. Bài báo cáo của dược sĩ Paul Pitt thì đặt cho chúng ta khá nhiều câu hỏi, là khi mà sử dụng đầu tiên đấy thì sử dụng progesterone là cần phải dùng trong trường hợp mà hỗ trợ sinh sản, thì có thể chúng ta dùng progesterone tiêm bắp thì 50 mg tiêm một lần trong một ngày, hoặc là tiêm dưới da 25 mg, hoặc là chúng ta dùng dạng gen thì 90 mg. Cái biệt dược của nó là crinol, hoặc là chúng ta dùng cái loại thuốc đặc như utrogestan thì 200 mg một lần, và cứ cách 8 giờ chúng ta đặt một lần. Như vậy là tổng liều của nó là 600 mg một ngày. Và kết luận của tiến sĩ Paul đấy thì Thụy Sĩ thì người ta không dùng về duphaston nữa, gần như cấm. Ở Pháp thì không dùng duphaston trong với bất kỳ cái trường hợp nào để mà giữ thai, vì người ta cho rằng là khi mà dùng như thế thì nó sẽ phải chuyển hóa qua gan này, tác dụng không nhiều này, bị phân hủy này, và thậm chí có người còn dẫn đến suy gan. Và suy gan vẫn là đang chờ để mà ghép gan thì cái thai đấy nó bị chết. Cái thai đấy chết đấy là những cái, tất nhiên đây là một quan điểm cá nhân chứ còn cũng không phải tất cả. Và chúng ta cũng nghe có những cái báo cáo thì rất ca ngợi về cái vi hạt này, và cứ uống thay luôn cả cái utrogestan, thay luôn cả ciproret. Nhưng mà cuối cùng thì đối với bài báo cáo của dược sĩ Paul ấy, thì cũng cho rằng là cái dùng cái progesterone dạng đặc âm đạo vẫn là tốt nhất vì mấy lẽ. Thứ nhất là gần cơ quan đích, đấy là gần tử cung, và đặc biệt là niêm mạc tử cung. Thứ hai là nồng độ progesterone trong máu nó sẽ duy trì được ổn định. Thứ ba là khi mà dùng thuốc đặc đấy nó dễ dùng nhất và ít bị kích ứng và không qua gan để mà bắt buộc gan là phải chuyển hóa, và ít tác dụng phụ. Và chắc là cái thuốc này thì hãng bên danh nhiều, đúng không? Thì cái kết luận này của ông Paul thì quan trọng. Và có một cái chúng ta thấy nữa, thì ông cho rằng là đối với một số trường hợp thì đặt cái progesterone liều rất cao, thậm chí có nghiên cứu của ông đặt đến liều là 1.200 mg trong một ngày, thì kết quả thai nghén tốt và cái tỷ lệ thai cho đến lúc sinh khỏe mạnh là cao. Và ông này thì đưa ra cho cái nồng độ trong máu nên duy trì là ở progesterone nồng độ là 12 nanogam trên ml, cao hơn so với tiến sĩ lúc đầu báo cáo là 10,64. Thì tất nhiên là những bài báo cáo nào thì chúng ta cũng là ghi nhận những cái những cái thông tin quan trọng. Đấy là những cái điểm mà chúng ta cần nhớ. Thì còn đối với giáo sư người Ý Sandro Gelli thì nói rằng là về cái làm hỗ trợ sinh sản thì cần phải chuẩn hóa mà mấy cái mà rất quan trọng, có tác động rất là mạnh mẽ đến cái kết quả của thụ tinh trong ống nghiệm, kết quả của hỗ trợ sinh sản, đấy là cái chất lượng phôi, rồi cái niêm mạc tử cung và kỹ thuật chuyển phôi, tức là yếu tố của con người mà làm là trực tiếp bác sĩ lâm sàng đấy. Thì đưa ra là có kinh nghiệm thì thấy rằng là thứ nhất là phải chuyển phôi ở dưới siêu âm, thứ hai là không dùng catheter cứng mà phải dùng catheter mềm, thứ ba là phải loại bỏ được cái chất nhầy ở kênh ống cổ tử cung, thứ năm là tốc độ bơm phôi, và người ta cho rằng tốc độ bơm phôi càng nhanh thì tỷ lệ có thai càng thấp, tốc độ bơm phôi càng chậm thì tỷ lệ mang thai được càng tốt, và cần phải có siêu âm hướng dẫn, và siêu âm hướng dẫn là siêu âm đường bụng là tốt nhất, và đầu catheter đến đâu thì dừng, thì cho rằng là đặt ở không chạm đáy tử cung nhưng mà đặt ở gần đáy tử cung thì nó tốt hơn, và nếu như mà đặt ở gần cổ tử cung, quá gần ở lỗ trong của cổ tử cung thì không tốt. Và người ta cho rằng khi mà theo dõi cái phôi sau khi đặt đấy, trong vòng 1 tiếng đồng hồ thì thấy có một số phôi di chuyển, còn có một số phôi thì hoàn toàn không di chuyển, đặt đâu thì nằm đấy. Và nếu như phôi mà di chuyển ngược về phía đáy tử cung thì tỷ lệ làm tổ và thành công cao hơn so với những cái phôi di chuyển về phía lỗ trong của cổ tử cung. Đấy là theo về nghiên cứu như thế. Và khi đặt được catheter vào rồi thì chờ bao lâu để rút ra, thì có nghiên cứu là chờ 30 phút sau trở lên, hoặc là có cái thì khi nhả phôi ra thì người ta rút catheter ra luôn, thì kết quả như nhau, không quan trọng. Nhưng khi mà rút catheter ra thì bao giờ cũng phải có động tác kiểm tra liệu catheter nó còn sót lại không, có dính vào cả phôi, còn dính lại catheter hay không, thì cái đấy là bắt buộc là phải làm. Và còn bệnh nhân sau khi chuyển phôi có cần nghỉ hay không, hay là dậy hay gì thì chẳng có gì quan trọng cả, cho bệnh nhân dậy, thậm chí đứng và đi lại cũng chẳng làm sao, mặc dù phôi có một số tỷ lệ phôi di chuyển nhưng mà không sao cả, tư thế tử cung đổ trước hay đổ sau cái gì đấy thì bệnh nhân nằm hay không thì cũng không có ý nghĩa. Về gặp một số các cái trường hợp khó khăn là cần phải nong cổ tử cung, tại vì khi mà gặp trường hợp chuyển phôi khó thì người ta khuyên là nong cổ tử cung ít nhất phải độ 2 tháng sau khi nong cổ tử cung rồi là chuyển phôi, mà cái thời gian để sau khi nong càng lâu càng tốt. Và thậm chí những trường hợp mà con nong cái gì cũng vô cùng khó thì cũng có kỹ thuật đâm kim xuyên qua thành của cơ tử cung mà chúng ta chuyển phôi. Thì cái này cái hồi mà tôi bác sĩ Phương Lan sang học ở bên Nhật thì một giáo sư Nhật người ta đã chuyển phôi như thế, tôi thấy cũng nhẹ nhàng thôi, và chúng ta đã làm được kỹ thuật giảm thiểu phôi thì tôi nghĩ rằng chắc chắn kim chúng ta cũng đâm xuyên để chúng ta chuyển được. Nhưng mà cũng rất may mắn, chẳng hạn như tôi thì chưa trường hợp nào mà tôi phải chuyển phôi bằng cách là đâm xuyên catheter qua cơ tử cung để mà luồn catheter qua kim để mà chuyển. Nhưng mà nếu như có khó khăn như thế chắc chúng ta cũng làm được chứ không phải là quá khó. Đấy là cái vấn đề về chuẩn hóa ở trong chuyển phôi, vì là nó cũng hết sức là quan trọng. Còn về chất lượng phôi bên niêm mạc tử cung thì chúng ta cũng là quan tâm nhiều rồi, nhưng về cái bản thân của bác sĩ lâm sàng để mà chuyển phôi đấy, vì nó có rất nhiều các yếu tố tác động. Nào là làm thế nào để không kích thích tử cung, lấy hết chất nhầy để đưa catheter vào nhẹ nhàng rút ra, người ta gọi là nhả phôi, nhè phôi ra, chứ không phải bơm phôi, có nghĩa là gì, càng chậm càng tốt. Thế còn về cái báo cáo cuối cùng của giáo sư Alberto Simeoni thì đây là giáo sư cũng không phải là chuyên ngành về hỗ trợ sinh sản, mà ông là thuộc về lĩnh vực chu sinh học, và ông cũng nói rất nhiều về cái trẻ em, về những cái bào thai, về nhĩ khoa, thì có mấy cái mà cũng là điểm qua. Đấy là nhất ấy, là đại đa số mà những cái đứa trẻ mà sinh ra từ IVF thì đều là khỏe mạnh, Tuy nhiên trong số đó cũng có một số các tỷ lệ trẻ bị bất thường, vì những dị tật, đặc biệt người ta chú ý đến dị tật tim mạch, dị tật thần kinh, dị tật về tiết niệu, như lỗ đái thấp, rồi dị tật về tiết niệu của nam giới, của là trẻ trai. Nhưng mà người ta cho rằng đấy là do những tác động, do những ảnh hưởng của di truyền của bố mẹ để lại. Nhưng một điểm để người ta nhớ là tác động của môi trường bên ngoài nhiều khi không kém phần quan trọng, thậm chí có khi còn mạnh hơn so với là di truyền, và cần can thiệp sớm. Có một nghiên cứu, người ta nghiên cứu đối với những cái đứa trẻ sinh ra từ IVF ấy, ở cái vị tuổi thiếu niên, thì thấy rằng những đứa trẻ này nó gặp tỷ lệ huyết áp cao rất sớm, mặc dù không phải là cao rất nhiều, nhưng mà cái tỷ lệ cao rất có ý nghĩa thống kê. Và cái này thì nó nghiên cứu thì thấy rằng là nó bị giảm cái giãn mạch này, rồi nó tăng cái sóng của mạch này, rồi là thành của động mạch dày lên này, và nó khác nhau giữa cái chênh lệch giữa tâm thu và tâm trương khác nhau rất có ý nghĩa thống kê. Và đấy là mang tính tim mạch của những cái đứa trẻ ở cái tuổi thiếu niên và sinh ra từ cái thụ tinh trong ống nghiệm. Và người ta cho rằng là những cái đứa trẻ mà sinh ra từ thụ tinh ống nghiệm này và nó có những cái dị tật như vậy ngoài tác động về di truyền của bố mẹ thì nó còn có những yếu tố ảnh hưởng, ví dụ như là sinh non này, do đa thai này, thai chậm phát triển trong tử cung này, rồi sinh non này, rồi viêm nhiễm này, và như vậy là và tác động của phôi nhiều, rồi là những cái thao tác ở trên phôi ấy, thì và cái môi trường nuôi cái phôi vân vân, rồi nó ảnh hưởng rất nhiều đến những cái dị tật, đến những cái bất thường của khi cái đứa trẻ sinh ra, ngoài cái rối loạn di truyền mà nó dẫn đến những cái dị tật bẩm sinh. Và chính vì vậy mà những cái đứa trẻ như sẽ sinh ra cái chuyện là nhẹ cân, mà là nhẹ cân này do sinh non, rồi do chậm phát triển, do người mẹ béo phì, do người mẹ bị bệnh tăng huyết áp, và đặc biệt những cái người mẹ mà dễ bị tiền sản giật và sản giật thì cũng dễ phải làm sinh non gì đấy, thì để lại cái hậu quả về tim mạch, về thần kinh, và nhất đối với những đứa trẻ sinh non nhẹ cân như vậy thì cái sự phát triển của hệ mạch máu của các cháu này thì rất là kém so với những cái đứa trẻ mà sinh nặng cân. Do đó là người ta xu hướng bây giờ chuyển phôi, thì chúng ta kết luận được ở cái chỗ này ra được cái bài học kinh nghiệm là không nên chuyển nhiều phôi, mà chúng ta chỉ chuyển đơn phôi để tạo ra được những cái thai chỉ là đơn thai, mà đơn thai thì nguy cơ sinh non là nó sẽ thấp hơn rất nhiều so với đa thai. Chúng ta qua bài học và kinh nghiệm của bài báo cáo của giáo sư chuyên về chuyên môn sinh học của là thụy sĩ là như vậy. Bây giờ chúng ta cũng có thể là thảo luận để chúng ta hỏi han thêm các cái báo cáo viên, mà không tôi cho rằng một buổi hội thảo mà tiếp nhận được chừng đấy kiến thức là cũng tốt lắm đấy. Cảm ơn Bê Danh nhé. Bê Danh ok.
Dạ vâng ạ. Dạ vâng ạ, xin cảm ơn phần tổng kết rất là bổ ích của giáo sư ạ. Hiện nay ban tổ chức cũng có ghi nhận một số những cái câu hỏi của các quý vị bác sĩ online gửi về cho ban tổ chức. Thì do thời lượng có hạn nên là ban tổ chức cũng xin phép để trả lời một số những câu hỏi trực tuyến ạ, để sau đó thì chúng ta có thể dành thời gian để thảo luận được ngay được ạ. Hiện tại thì có một câu hỏi của bác sĩ Thư gửi về ạ. Xin giáo sư chia sẻ kinh nghiệm tại viện về việc đo nồng độ progesterone vào ngày chuyển phôi. Kết quả sẽ dự đoán được trên lâm sàng thực tế là như thế nào ạ?
Vâng, như lúc nãy tôi cũng đã đặt vấn đề và nói đấy, hầu như là chúng ta chưa định lượng progesterone trước cái ngày chuyển phôi. Chúng ta chỉ định lượng progesterone khi mà cái ngày lấy noãn ấy, thì nếu như nồng độ progesterone cao ấy thì chúng ta cho đấy là nó hoàn thể hóa sớm gì thì chúng ta không chuyển phôi. Chứ còn đặc biệt là những cái trường hợp mà chuyển phôi trữ thì chúng ta là chưa làm cái định lượng progesterone. Còn sau hội thảo này thì tôi khuyên mọi người ta nên định lượng progesterone, tại vì hầu hết tất cả các cái phòng xét nghiệm của các trung tâm hỗ trợ sinh sản nào cũng đều định lượng được progesterone. Chúng ta định lượng thử cái xem, tại vì chúng ta cũng gặp một số các trường hợp là chuyển chúng ta chuyển rất tốt, đặt phôi vào rất đúng vị trí, phôi hình ảnh thì rất là đẹp, nhưng mà chả hiểu làm sao mà nó cũng chẳng có thai, hay chăng là do cái nồng độ progesterone khi cái ngày chuyển phôi đấy nó dưới 10,6. Thì bây giờ chúng ta nên xét nghiệm. Và tôi cho rằng là lúc nãy thì tiến sĩ có báo cáo là nếu như mà nồng độ progesterone mà nó giảm dưới 10,64 đó, đúng không? Thì chúng ta nên tiếp tục đặt thuốc, rồi sau đấy để định lượng lại. Mà theo tôi mà chúng ta đã định cái ngày mà chuyển phôi rồi, mà cái ngày đấy chúng ta định lượng progesterone thấy nó giảm dưới đấy thì chúng ta cancel và chúng ta chờ chu kỳ sau. Chứ chúng ta mà cứ tiếp tục đặt mà thì có khi đang đặt thuốc nhưng nó vẫn ra kinh đấy. Do là không nên làm cái đấy, nhưng mà chúng ta nên đặt. Còn cái vai trò của progesterone thì hết sức là rõ rồi, mà cái này là từ những cái cuốn sách rất là kỹ rồi, và đã là một người giáo sư ông Mackey, ông đã là thử nghiệm và là đánh giá cái vai trò của progesterone đối với niêm mạc tử cung. Ông đã là lấy một cái mảnh niêm mạc tử cung cấy vào tiền phòng của mắt khỉ đấy, rồi cũng cho cứ tiêm progesterone thì cái mảnh cái niêm mạc đấy nó vẫn giữ nguyên, nhưng mà khi ngừng cái tiêm progesterone do tụt progesterone thì cái mảnh niêm mạc mà cấy vào tiền phòng mắt khỉ nó bị bóng do là không có progesterone và nó chảy máu giống như chảy máu kinh, và người ta cấy vào tiền phòng vì ở chỗ cái tiền phòng của mắt khỉ quan sát rất dễ qua.
Dạ vâng ạ, xin cảm ơn phần trả lời rất bổ ích của giáo sư ạ. Kính thưa quý vị bác sĩ, em biết là vẫn còn rất nhiều câu hỏi được gửi về cho ban tổ chức. Tuy nhiên thì vì thời lượng hội thảo trực tuyến có hạn nên là ban tổ chức cũng xin phép được ghi nhận các câu hỏi và gửi về cho báo cáo viên trả lời cho quý vị bác sĩ sau. Riêng phần thảo luận online thì chúng em xin phép được kết thúc tại đây, và quý bác sĩ vui lòng dành chút thời gian để vào link khảo sát trả lời câu hỏi để nhận video record và slide tài liệu nếu có nhu cầu, và ebook sau hội nghị, hoặc có thể quét mã QR code trên màn hình để nhận tài liệu ạ. Thay mặt Bệnh viện Phụ sản Thiện An cùng văn phòng đại diện Beijing Healthcare xin được gửi lời chúc sức khỏe đến toàn thể các quý vị bác sĩ đã tham dự nhiệt tình, và mong rằng trong thời gian gần nhất sẽ có nhiều cơ hội được đồng hành với quý bác sĩ nhiều hơn. Xin được trân trọng cảm ơn. Xin được kính chào và hẹn gặp lại ạ.
Một tay hội thảo thành công quá nhỉ.